Hey everybody, my name is Adam Akkad, and I'm teamed up with Brycelyn Whitman, and we're presenting this week, first week on a journal article called Scarless Genome Editing for Human Pluripotent Stem Cells via Translumin Primomycin Selection. So let's just go ahead and introduce a very important topic that's discussed throughout the paper, and that's CRISPR Cas9. It's the primary methodology they use for the genome editing in the paper, and it's really important that we understand all of the attributes of this system. So basically, CRISPR Cas9 is a bacterial defense mechanism that is used in bacteria to fight off viral infections. But we've adapted it and seen that it can actually be very useful for specific gene editing. The main components, this is just a review slide from the slide we had in the lecture. So the main components is the Cas9 protein, which is an endonuclease, and a single guide RNA that has two parts, a cRNA and a tracer RNA. The cRNA is complementary to the target of interest, where the endonuclease cuts the gene in the genome. So this is about 20 base pairs or nucleotides long, and this is where the CRISPR part comes in. This is a clustered regular interspaced short palindromic sequence, which is a palindromic sequence that's repeated and basically short, that's going to be removed from the gene of interest and replaced by a double-stranded blunt-ended break. And you also have the tracer RNA, which is an enzymatic and structural component of the single guide RNA that links everything to the Cas9 protein. And you have these short PAM sequences that need to be present in the genome before splicing can occur. And these are actually these are found in viral genomes, and that's kind of indicative of the immune defense mechanism. These are remembered, so quote unquote, by bacterial cells. So the main takeaway is that CRISPR-Cas9 induces blunt end double-stranded breaks at specific locations in the genome. That's the main function of this mechanism. So in terms of the repair mechanisms after this double-stranded break is induced, what does it look like? What do we have in the cell to fix these? So we have two primary methods, which is NHEJ, which is a non-homologous end joining, which is also known as an insertion or deletion of a random gene. Insertion, that is, insertion of a random sequence. Not gene, it's random sequence. And we have HDR, which is a homology-directed repair. So this should come off as a review also. I found these figures online from helpful resources. So in terms of insertion deletion, as it sounds, you have a double-stranded break, and they're either joined right at that break, or a random non-homologous sequence is inserted into that between the two broken segments off where that break was. In terms of homology directed repair, HDR is what we want. This is the ideal resolution from this break, although it is a rare event in the cell. Regardless, HDR uses a template strand or a template DNA fragment to fix the double-stranded break and basically replace the double-stranded break with a specific sequence. In the cell, it would be usually a sister chromatid that has a similar sequence, although a different allele, that would use the template strand to fix and repair this break. But we can kind of hijack this system, and instead of having a natural repair, you can use this mechanism to copy or repair the break with a strand of a fragment of DNA or template DNA that we introduce, that comes from us, which is the whole gene editing aspect, a new gene in that place at that specific location cleaved by Cas9 and CRISPR Cas9. So that's HDR, and this will be heavily talked about in the paper as we go forward. So with that background information in mind and that knowledge, what is the objective of the study? Well, first off, the paper mentions and really makes it apparent that traditional CRISPR, as it's done, is inefficient and scarring. What do I mean by scarring? Basically, because of the way that the template DNA is introduced and the CRISPR Cas9 mechanic DNA is introduced in the cell, the cell is, after the process is done, the genome is left with inserted or deleted sequences or scars in the genome, throughout the genome, that are unwanted, that are not the specific gene editing or gene insertion or sequence insertion of whatever we wanted it to initially be, and that is considered a scar, and it's unwanted. And the objective of the study is to kind of get rid of these problems. So the objectives is to increase the efficiency for screening of gene edited clones, and increase the ratio of HDR to NHEJ, which is, as we talked about, the more desired outcome of double-stranded break repair, and also mainly to prevent scarring. So they do this, but first, before they actually go ahead and do this, they explain kind of literature that's been done and different methods that have been done in the past to kind of mitigate each problem, or kind of improve or tap into or answer each of those objectives separately. One example is increasing the efficiency of screening for edited clones, meaning looking at or understanding and screening the result of the edited genome, or which genomes are selected for which cells have actually been edited properly. So this one paper discusses a method of a non-antibiotic selection technique that selects for mutant and non-mutant genes in cells with fluorescent tags, and they use this TALEN sequence that finds the mutant and non-mutant sequences and allows for binding for a fluorescent probe. And this process basically improves mutant selection and screening as opposed to traditional methods. Although this isn't a process that they use in the paper, it was an interesting paper that they cited and build on. So in terms of how they're going to get rid of scarring, they cited other papers mainly that specifically used short single-stranded oligonucleotide donors as their DNA fragment that houses the genetic information for CRISPR Cas9 proteins and the selection or resistance genes. So basically what these are single-stranded DNA that can be in plasmid or linear that are introduced in the cell, and the outcome is that they have been observed to not produce scars through CRISPR Cas9 editing. And the reason they do this, they hypothesize many of these papers, is that it's unlikely that a single-stranded DNA will be directly or randomly introduced into the genome, unlike a double-stranded fragment or a plasmid that has been observed to do so. So the main takeaway is that instead of a double-stranded template that's susceptible to this random genome integration, a scarless single-stranded short donor is used as a template. So what is the main hypothesis of after going through extensive review for all these different potential solutions to these problems? The main hypothesis of this paper is that a stringent selection, i.e., using puromycin selection, for cells expressing high levels of Cas9 will both increase rates of gene knockout and also increase rates of HDR when delivered with a short single-stranded DNA donor. So ultimately, they introduce a new method using CRISPR Cas9 with many different improvements that improve the overall gene editing efficiency. The ratio improves the ratio of HDR to indel, and makes the whole process Scarless. So let's get straight to the figures. The first figure is basically a schematic of the SSODN that is used to make this process more efficient, to answer that main objective of making the whole process more efficient. They use a plasmid, a single-stranded DNA plasmid, that houses both the genetic information for the sgRNA as well as the CRISPR Cas9 protein and the resistance gene. So the fact that they're both in the same plasmid that's one form of making the whole process more efficient, because they don't have to be in separate plasmid and getting transplanted at different locations. So another main feature of this SSODN is the 2A sequence, which is right smack in the middle between the resistance gene and the Cas9 protein gene. That is a skip sequence, which means, so this is DNA, right? This plasmid is DNA, which means it's going to be transcribed and then, or reverse transcribed, and then translated into a protein, a peptide, or a protein. And so the 2A sequence in the RNA will allow as it sounds, the ribosome to basically skip over that sequence and release the Cas9 protein that's been made before it goes on and starts translating the rest of the sequence. This is important because it makes the process more efficient. Again, it keeps the ribosome from disassembling entirely before it starts translating the resistance gene, which is a notable improvement to efficiency. So what about figure 1b? This is the second part of the figure. After they introduce that SSODN, they look at puromycin selection, which is that stringent selection that they were talking about in the hypothesis. And so B here is a timeline of events for introduction of the different puromycin concentrations and ROCK inhibitors to allow for basic cell culture of these iPSCs. But they also introduced the plasmid. The plasmid, so in minus, the plasmid is not introduced, and plus, the plasmid is introduced. And in both cases, puromycin is introduced over time. Just a quick side note and review, they introduced the plasmid via electroporation, and as we know from the lecture, electroporation is a process in which an electrical pulse is induced to create pores temporarily in the membrane to allow for things to pass. Specifically, the electrical field allows for the negatively charged DNA molecules to pass through, i.e. the plasmid, to pass through the membrane. And from there, it's further localized into the nucleus, the plasmid itself. So with that in mind, it's seen here on the part C of the figure that after 12 days, a significant colony is observed in the cells with plasmid, versus no colonies are really observed in that without.